Good morning, everyone, and welcome to our session today covering irrigation research and management strategies. And I'm really excited to bring to you two great speakers, and we'll get to them in just a moment. I just wanted to sort of set the stage for why we're talking about this. And aside from the obvious kind of context that we hear every day about climate change and things getting warmer and drier and things like that. The fifth Oregon climate assessment was actually just released in January of 2021. And you can find the citation for it here at the bottom of the slide. And so you can punch that in. And it's a really thorough document that sort of highlights all of the up-to-date research on climate change that's been going on, and it's run by this Oregon Climate Change Research Institute out of Oregon State University. Really nice document. So just to set the stage here and give you all a little bit of a context, some of the information that they present. Um, you know, it's no secret, I think, at this point that the trends for annual mean temperature have been increasing for some time, and on the left you'll see a figure that highlights that, and you can see the break in that gray area on the left hand side. That's sort of the observed what we've seen, and going to about 2020 or so, and or maybe a little bit before that, I believe, the gray part. But in any case, on the right hand side you'll see the orange and red shaded areas and the lines that sort of describe the averages, and the shaded areas are the sort of variation around those averages. But you can see that the annual mean temperature trends are certainly projected to go up and to increase. And what they write is that we're expecting about a 5-degree Fahrenheit increase by the 2050s, and maybe a potentially 8.2 degrees Fahrenheit annual mean temperature increase by the 2080s. And for the longest time, I've been looking at all these dates like they're so far in the future. But if you consider that a production vineyard has a working lifespan of about 30 years, those vineyards that you're maybe planting this year uh, potentially will be experiencing these higher temperatures by the end of their lifespan. So certainly not that far away. The story with precipitation is a little bit more confusing or variable, and they highlight that. But what they point out is that despite the fact that the variability in precipitation going forward into the future in Oregon is going to remain, what we're expected to see is that we're going to see a larger proportion of precipitation falling as rain rather than snow, largely due to the increased annual temperatures. And that's going to increase, that proportion of rain versus snow is going to increase as we go into the future, particularly at the lower elevations. Now that's important, again, because the snowpack is our largest water storage reservoir. Extreme heat events is something that we're always concerned about in grape growing, and they highlight some trends and projections as well. And over here on the left-hand side, you'll see the historic trends from 71 to 2000 of the number of days in the growing season, April through October, with a heat index greater than 90 degrees Fahrenheit. And you can see that we have more hot days, of course, in Southern Oregon and out here in the Eastern Oregon and Snake River valleys. Not so many extreme heat events in the Willamette Valley here. But as you move forward into the future, and these projections cover 2040 to 2069, you can see that a really large number of days, up into the 60, 70, and 80 days, with a heat index above 90 in Southern Oregon. Even in the Willamette Valley, we're looking at maybe 30 to 40 days of very, very extreme heat events. So definitely something to be concerned about going forward. And what they write, I'll let you read this. I'll leave it up as I speak here. They point out that the frequency and duration of these extreme heat events is going to increase, and the summers are going to be warming faster than the winters. And they point out here, and the bolding is mine, that the hot summers of 2015 and 2018 are salient examples of summer temperatures that are expected to become relatively common by the middle of the 21st century. So for those of you who remember the summers of 2015 and 2018, very warm summers, expect that to be the new normal, is what they're writing in this new climate assessment. And lastly, of course, drought is related to all of this. And this is a nice figure, although it's very colorful. And so I do apologize about that. We've all seen the drought monitor before, with the picture of the state of Oregon and various shadings of yellow, brown, and red. And those same colors are represented here and relate to the same data here on the bottom of the figure. And then we have their sort of inverse here on the wet side on the top. And this is just a figure showing historical trends of what's called the Standardized Precipitation Evaporation Index. And it's essentially an index that compares the total precipitation and the total ET. And it's a really useful index. And they write that it's a key quantitative metric for assessing occurrence and severity of meteorological and hydrological drought. And what you can see is there's all sorts of different years that are drier and wetter as we move through time, as we approach modern times. But you can see that the most recent years have been quite dry, similar to what we saw in the 20s and 30s. And those, of course, were the Dust Bowl years. And what they write is, as summers in Oregon continue to become drier and warmer, the mountain snowpack is going to decrease. The frequency of droughts related to that decreased snowpacks are going to increase. And again, they highlight that 2014 and 2015 are good sort of marker years to look at in terms of what we might expect more regularly going forward. So with all that in mind, we certainly have the tools to manage through this, of course, providing that we have the supply of water. But what we really are trying to understand is we really want to know and improve our understanding of how grapevines respond to extreme heat events and drought. Once we know how grapevines respond to these types of environmental hazards, you could say, physiologically, then we might be able to implement some sort of management strategy. And so the question then is, what can we do about it in the vineyard? So we're going to have two speakers today that are going to cover some of that. The first speaker, Dr. Simone Castellarin. He is a good friend of mine, and he's the Associate Director of the Wine Research Center at the University of British Columbia. And he is also Associate Professor at the Faculty of Land and Food Systems at the University of British Columbia. And Simone got his PhD at the University of Udine in Italy, way back in 2007, I think it was. And his research program primarily is involved in investigating biological mechanisms that determine grape and wine quality with a focus on ripening processes. And he's really interested in studying how grape quality is affected by environmental factors, namely temperature and water availability, and developing viticultural strategies that can deal with some of these challenges that we experience. And I want to point out that in between his PhD and his professorship, Simone was a visiting scholar at UC Davis a little bit before I got there. But not only is he an accomplished scientist, but he was quite the home gardener as well. In fact, when he was a visiting scholar at UC Davis back in 2008, he was recognized by the local paper for his pumpkin growing ability. And he got a nice little write-up in the local paper here about how he won the pumpkin growing contest at UC Davis. So I just want to highlight that he's a Renaissance man. After Simone, we have Dr. Nazareth Torres. She is a postdoctoral scholar at the Department of Viticulture and Enology at UC Davis. Dr. Torres received her PhD from the University of Navarra in Spain, and she was studying the influence of mycorrhizal inoculation on phenolic composition and antioxidant capacity of grapevine, specifically Tempranillo, under different climate change scenarios, and she won an award for her doctorate. Currently, her expertise is in the fields of irrigation management, soil health, arbuscular mycorrhizal fungi, and grapevine symbiosis in the context of climate change mitigation. And so what we'll have, I think, is that Simone is going to give us a nice little overview of water deficit impacts on grapes and highlight a few different studies that he's been a part of. And Dr. Torres is going to give us an overview of a recent study that she just completed about management strategies for drought and grapevine and how the drought impacts carbon partitioning. So with that, I will stop sharing.
Right. So good morning, everybody. And I would like to thank the organizing committee for the invitation and also the chair of this session for the nice introduction. So my talk today will be about the impact of deficit irrigation strategies on the grape quality. We'll also talk a bit about the impact on the wine quality side. So as it has been introduced, we expect climate to change, is changing, and we expect actually an impact of this change on viticulture. This is a study from 7 years ago where scientists have shown in this map with red colors the current area where we produce most of the grapes and the wine in the world. And they forecasted that there will be an expansion of those areas to mostly to northern regions of Europe and North America, and where we expect to be able to produce grapes and wine in the next probably 50 years. Well, as it has been introduced in this session, we also expect higher temperatures in the current viticultural regions, as well as more frequent drought periods during the summertime. The topic of how to manage drought in vineyards has been actually investigated for many years now, and it has been always like a hot topic. We know we do need water to manage grape production, but we also know that we have to tune water carefully because by applying water, we might affect the quality of the grapes and also the quality of the wine. So we want to optimize the use of water in vineyards to optimize the quality of fruit and wine. And we know that grapes are pretty adapted to dry environments. Here is a nice vineyard from Spain, from the Canary Islands, where growers grow grapes with no irrigation, with just 120, 150 millimeters of rain. I will say 40, 50 inches of water per year. But obviously you see that these low amount of water strongly limit the growth of the plant. And as you can see here, the yield is also severely affected. So these type of productions are not always economically sustainable. In a similar scenario in British Columbia, Canada, we have in the Okanagan Valley, one of the major wine regions in Canada, we have about 40, 50 inches of rain or snow per year. But we have the availability of irrigation water from the Okanagan lakes and other lakes that are in the surrounding areas. And we can basically grow full canopies and produce yields that allow the production to be economically sustainable. Well, managing water and actually managing water deficit is, as I said, an important topic, and is still an important topic, despite the fact that we have been studying it, and we have been studying the physiological response of the grapevine to drought and to water deficit for several years now. We do know that drought affect leaf gas exchanges. The drought limit transpiration and limit the photosynthesis of the grapevine. It limits the canopy growth, as we have seen from the previous pictures, and also limits the yield. But we know that drought and water deficit might affect and have direct effects on fruit composition. And therefore, it can affect the quality of the wines. So it's important to manage this drought to optimize quality of grapes and wines. Well, part of I would like to start my talk by introducing some of the major compounds that affect the quality of grapes and wines. And these compounds are aromas and phenolics. I'm sure that the audience here is very familiar with several of these compounds. In this group of compounds, we have aromas like terpenes that affect the aroma of Riesling, Gewürztraminer, Muscat. And then we have other types of aromas like methoxypyrazines that are more well known for affecting the quality of Cabernet Sauvignon, Cabernet Franc, as well as we have phenolic compounds that are anthocyanins, the red pigments, or tannins that affect astringency and bitterness of the wine. Actually, most of these compounds are synthesized in the skin of the berry. And for this reason, when we reduce or increase the size of the berries by managing irrigation, we can affect the concentration of these compounds in the grapes and later on affect the concentration in the wine. Basically, what we know is that when we have smaller berries, we tend to have a higher concentration of these compounds because they are synthesized in the skin, and we have less flesh and less water, and so we have a higher concentration of those aromas and those phenolics. Vice versa, when we have more water, we might dilute some of these compounds by increasing the amount of water in the flesh. And this can occur via, irrigation, but also can occur in those seasons where we have high precipitations. This is why growers and winemakers often cared about having small or smaller berries in vineyards to have higher concentration of those important quality compounds. And studies that relate to water deficit and of some water deficit during the season in vineyards started actually in the mid-80s when Dr. Matthews and colleagues at UC Davis applied different deficit treatments at different times of the season to Cabernet Franc and Cabernet Sauvignon grapevines. Here is the results of one of those studies where they shown that by applying some level of deficit before variation or after variation, or prolonged levels of deficit during the season, they were decreasing the yield, but at the same time they were increasing the concentration of anthocyanins or the concentration of total phenols. This was also having an effect on the sensory of the grapes and sensory of the wines. We did ourselves some of these studies in the past years, and we worked mostly on Merlot as a variety, and some of those experiments were conducted in North Italy. And we did similar studies where we were comparing well-irrigated grapevines with grapevines that were exposed to severe water deficit. And basically, we were withholding irrigation during most of the season and analyzing what were the effects on the composition of the grapes and the composition later on of the wine, and we had quite consistent results with what was already found by Dr. Matthews and colleagues, and other studies that later on were performed. So when we apply some level of stress or water stress in our vineyard, we can increase the concentration of pigments or anthocyanins and the concentration of tannins as well. But we also observe in our studies that it was not just an effect of reducing the size of the berry via the application of water deficit, but we found out that water deficit was also directly affecting the biosynthesis of these compounds. We performed several molecular studies to figure this out, but the take-home message of those studies is, yes, by applying water deficit or let's say drought in vineyards, increase the concentration of pigments and tannins, not only by reducing the size of the berry, but also by stimulating the biosynthesis of these compounds. And we also observed in this study that several aromas were affected, and we observed that in the wines produced from those grapes, the sensory feature did change. So there was the wines that were produced from vines that were exposed to deficit had a darker color or a higher intensity of the color, of the red color, and the wines had also had a higher intensity of those aromas associated to red fruits or also to jam or jam-like aromas. So we can, by apply severe water deficit on red grape varieties, basically increase the pigmentation, affects also the aromas toward more ripe type aromas rather than green type aromas. It was less known, though, the effect of drought or water deficit application on white grapes. It's not very common that we apply water deficit in vineyards on white grape varieties. And we started conducting some studies in 2012, where we took a white grape variety grown in North Italy called Tokai Friulano. And we, as you can see from this picture, well irrigated the plants, so we had a perfect, fully photosynthesizing canopy, and we applied. We stopped irrigating from fruit set on, and we exposed those vines to very severe water stress. And we look into the composition of the fruit and into the composition of the wines. An interesting result was that we observed that, particularly at harvest time, at the end of our experiment, the fruit harvested from those stress grapevines had higher concentration of terpenes. Terpenes are important, as I said before, volatiles in aromas that affect the quality of many important wines like Riesling and Gewürztraminer, just for example. So we observe a higher concentration of terpenes where the plant was subjected to water deficit. And we also observed that, related to the terpene biosynthesis were actually stimulated. So what I mean here is that, as we have seen for the anthocyanins, the metabolic pathways that were synthesizing terpenes, these aromas, were actually stimulated by water deficit, but by drought. That means that it was not just an effect on the berry size, it was more an effect on the biosynthesis, on so the metabolic pathways that are working during grape berry ripening. Well, this was also translated into higher levels of these compounds on this aroma in the wines. These aromas can be free, so volatiles, or can be what we call bound, so linked to sugars that make them not volatiles, but then later on they can become volatiles during fermentation or wine aging when this sugar is removed for the compound from the compound, and the compound becomes volatile. So our study showed that what we have seen on the grapes was also seen on the wine, and then by applying water deficit, we could increase the concentration of these important aromas. That was a positive result, but as for the Merlot study, those were kind of experiments where we applied levels of deficit that were very severe, and they were limiting severely the yield of our vineyard. So we had decreases in the yield of 30, 40, 50%, depending on the season. Not always those levels of stress are sustainable in commercial vineyards, obviously. So we moved on, and but the other point I forgot to mention, is that we were also very interested about these results because, as I said, these aromas are major constituent of many white wines and white grapes, and confer flowery and fruity notes to those grapes and wine. So we thought we wanted to follow on these studies to learn if we can develop strategies to apply that implies the application of water stress, but to levels that do not limit severely the yield, but can improve the composition of the fruit, for instance, by improving the production of these interesting aroma that might have a strong impact on the quality of many white grapes. This is how we came to this study that I will present briefly at the end of my talk. This study was conducted in the Okanagan Valley, British Columbia, one of the major wine regions of Canada, and in a region that the Osoyoos First Nations called the Inkamip Desert. So it's not actually a desert, it's a semi-arid shrubland, but it's a region where we have very low precipitations, 100, 130 millimeters of rain per year, 40, 50 inches of rain actually during the growing season. And in this region, we need water, we need irrigation to grow commercially grapes. But at the same time, we thought, well, let's see if we can develop some strategies to limit irrigation, apply some moderate stress levels to the grapevine so we can improve the aromas, save some water. So this is how we came with this study on regulated depth irrigation. So regulated depth irrigation means basically applying, managing irrigation so that our plant is under stress, but is under a regulated stress. And in our case, we decided to have a moderate stress that we knew was not going to have a strong effect on the growth of the plant and on the yield of the plant. But we thought it could have an effect on the quality of the grapes. This study was performed on Gewürztraminer, that is a variety known for its aroma. It was performed in a vineyard where you can see usually has 1600 growing degree days expressed in degrees Celsius, but basically not a cool area. It's a relatively warm area of Canada. And as you can see from the graph on the bottom, basically we in these vineyards we had we had precipitation up to June and July, and then from July normally to October we have no rain. So we have to sustain with frequent irrigation the growth of the grapes. Normally we irrigate once or twice per week. We performed the study for 3 years, 2016, 17, and 18. So the treatments we studied were the commercial, the standard irrigation treatment, that is the controlled treatment. This is what growers in that vineyard were doing. They were basically irrigated so the plant was not under stress. And then we had treatments that had a moderate level of water stress early on before veraison, late, meaning from veraison to harvest, and prolonged. Prolonged means we apply stress from 30 days after anthesis to harvest. So it was a prolonged water stress. And in this vineyard, what we did was to analyze leaf gas exchanges, photosynthesis, all the physiological parameters related to the grapevine, and then compositional parameters related to the fruit, like sugars, acids, but particularly aromas, so free and bound terpenes, the compounds I mentioned before. What I would like to add, though, is that we carefully manage irrigation by weekly measuring leaf water potential and by applying irrigation volumes accordingly. So basically, for our moderate deficit irrigation treatment, we were not irrigated until the leaf water potential was minus 1.3 megapascal, so 13 bars. Vice versa, for control, we were irrigating to keep the water potential minus 0.8 megapascal or 8 bars. And you don't have to focus on this graph, but basically the irrigation volume change, obviously, depending on the treatment. And with the early deficit, we could save 30% of the irrigation water. With late deficit, we saved 38% of the irrigation water. And with prolonged deficit, we saved 50% of the irrigation water. But now, how our treatments affected the growth of the plant, the physiology, the yield, and the composition of the grapes. Well, when we apply some level of stress, one of the first responses is to reduce leaf gas exchanges, so basically reduce the photosynthesis. And our stress level, despite we consider them moderate, reduced photosynthesis by 50%. So it was pretty consistent across the seasons. But our focus, again, was at harvest time. We wanted to know what was happening on the fruit and the composition of the fruit, but also what was happening on the yield, how our treatments affected the yield and the compositional components. So the first table that I'll summarize briefly is the one on the yield. So our treatments, some of our treatments did reduce yield, and they were our early deficit and prolonged deficit treatment. But interestingly, one treatment, the late deficit treatment, did not affect the did not affect yield. So by applying deficit later on during the ripening process, we did not affect the amount of grapes and the quantity of grapes we were harvesting. Then we measured the composition, total soluble solids, so the bricks and the tratable acidity. And out of our irrigation treatments, the one that actually had an effect was our prolonged deficit. The prolonged deficit reduced sugars and increased acidity in Gewürztraminer when compared to the controls. The other two treatments, early deficit and late deficit, have not had a significant effect on the composition as regards sugars and acidity. And now we go to the focus of our study. Our study was focusing on the effect of these treatments on terpenes, so on the aromas of the grapes. And that was an interesting result again. So one of our treatments, the late deficit one, consistently across the 3 seasons, improve the concentration of some specific terpenes like geraniol, citronellol, and a couple of other terpenes that are actually quite important, very important, some of the key ones for the aroma of these grapes. So the conclusion was that, yes, when we apply regulated deficit irrigation, we can actually increase the concentration of some of the free terpenes, and particularly geraniol, that is the main terpene that is synthesized in Gewürztraminer grapes. We also look at the bound terpenes, and the bound terpenes are the ones that I say they are not volatile, they are attached to a sugar, but they can become volatile during fermentation when the sugar can be removed by some enzymes that the yeast produce. Actually, despite there was a trend in the higher concentration, we never observed a significant increase of these bound terpenes under regulated deficit irrigation. So the result was that we had no significant effect of our treatments on the bound terpenes. And this leads to our conclusion, or the conclusion of my talk is that when the grape responds to drought, to severe water deficit, by increasing the synthesis of several compounds that are important for quality. I mentioned anthocyanins, I did mention aromatics. But severe water deficit is not always applicable to commercial vineyards. So we have developed, or we are developing now, some of these strategies that we call regulated deficit irrigation, where we apply moderate water deficit to vines at specific developmental stages. And we have learned from this study in Gewürztraminer that if we apply a moderate water deficit during ripening, we can save water. We save 38% of the irrigation water compared to the standard irrigation we apply to the same variety in the Okanagan Valley. But we can, by doing so, also increase the aromas of the grapes. And this is actually an interesting result because it tells us that we can save some water, and by saving water, we can modulate quality, grape, and potentially wine quality also in white grape varieties, not only as we traditionally thought in the past with red grape varieties, where anthocyanins and tannins are the major quality compounds. Right, this is it for my talk, and I would like to thank several people that have worked on this project, and also the industry partners at Andrew Peller and at MRS Vineyard Services that collaborated with us on this project, the funding agencies, the BC Wine and Grape Council, MITAX, and BC Investment Agriculture Foundation. But particularly, I would like to thank Eugene Kovalenko, the student that has performed the majority of this study, that is now working for the industry. And thank you very much to you for your attention.
So thank you so much to the organizer and to Ali for his nice invitation and presentation. As he said, my name is Nazaret Torres, and I'm going to present this talk entitled Consideration of Sustainable Irrigation in a Water-Scarce Region Under Warming Conditions. According to the California Water Resources, irrigate agriculture in California is the largest consumer of water, accounting for about the 80% of water supplied to the state. So water resources can reach a critical state due to the extended drought periods and the overuse by irrigated agriculture. So in this context, the irrigation of vineyards is extensively increasing in historically non-irrigated areas to mitigate warming trends, as well as many of the viticulture areas on the world relying on irrigation to consistent production, such as California, where irrigation is required to overcome the dry and warm summers. For example, a simple example is this one. In California, California uses 318 gallons of water just for irrigation per gallon of wine. And in 2018, 716 million of gallons of wine were produced. So I don't need to do the calculations to say to you like it's an important environmental issue. Because also the Californian climate is changing to little to no cloud cover with intermittent heat spikes, a general warming trend with no increase in precipitation supply. Actually, last February, the February of last year, was the driest February since there are records. Because of that, currently wine growers are aware of the importance of a sustainable viticulture to ensure the sustainability of the viticulture in the future. So irrigation optimization will still be one of the most desirable tools to improve crop productivity and quality. In this context, the aims of this study were to evaluate if the standard irrigation practices are economically sustainable in a warming climate, to investigate the effect of different irrigation amounts on the plant physiology and berry quality of Cabernet Sauvignon grown in Napa Valley, and to evaluate the impact of these irrigation practices on water resources and arbuscular mycorrhizal fungi associated with grapevines. To reach these aims, a 2 years study was conducted at the UC Davis Oakville facilities. The vineyard was planted in bale soil, loam soil, on Cabernet Sauvignon, grafted onto 110 rootstock, and spacing 6 feet spaced by the vines by 6 feet, in 9 rows. And between the rows, the space was about 7 feet in an oriented northeast-southwest. The vines were spur-pruned into a bilateral cordon on a 30 degrees open trellis system at a height of 36 inches. And then the canopy was vertically subpositioned at 45, 60, and 70 inches to favor the light penetration. The experimental design was a randomized flock with a one-way arrangement, and the irrigation treatments were based on in the follow explanation. So they were based in different fractions of the crop evapotranspiration. So the crop evapotranspiration was calculated using the following equation, where the ETC is equal to the ETO, the crop, the reference evapotranspiration, times the Kc. the crop coefficient. The application of the irrigation treatments were started in April of each year and to harvest. And the irrigation treatments were obtained by changing the emitted output. The calculation of the ETC was made with the 100% ETC, and then just varying the emitted output from 2 gallons to 1 gallon or half gallon, we get the other irrigation treatments, the 50% ETC and the 25% ETC, as a fraction of the 100% ETC. During the experiment, several variables were monitored. Between the grapevine, we measure the grapevine water status and the gas exchange parameters, the photosynthetic rates. Also, the vegetative growth and sugar metabolism, recording the leaf, shoot, trunk, and root biomasses, as well as the soluble sugars and starch contents in those tissues. Also, we recorded the yield components, and we analyzed the berry composition, primary and secondary metabolites. And finally, we also assess an environmental the environmental impact of these practices, studying the water footprint and the arbuscular mycorrhizal fungi abundance in roots. So here you have the results. As you know, 2020 was a extremely arid season compared to 2019 growing season, which was actually really rainy. And in 2020, the precipitation was like about 31 inches less of precipitation, while the temperature was quite similar, but we have like 70 days with temperatures over the 87 Fahrenheit. Here we can see the stem water potential of the plants. In all the graphs that I'm going to show you, the 100% ETC treatment is gonna be black. The 50% is going to be gray, and the white for the 25% ETC. As you can see, the stem water potential was decreasing along both growing seasons. However, in the second growing season, we didn't find any difference between treatments. On the other hand, the 100% ETC treatment improved plant grapevine water status in 2019 compared to the other two irrigation treatments. Regarding the carbon assimilation, we saw that 100% ETC treatment improved the assimilation rates over the two seasons compared to the other irrigation treatments. We measure, as I explained before, the vegetative growth of the plants, where was improved by the 100% ETC. As you can see, we record increased leaves, shoots, and roots biomass. But regarding the primary metabolism on these tissues, we just recorded and improved total soluble sugars and starch in the leaves. And no differences between treatments were recorded in the contents of these carbohydrates in other tissues. As you can see in the image, yield components were highly affected by treatments. And we recorded like the 100% ETC improved the yield and also increase the leaf area to free radius, increasing the canopies. While the 25% ETC decreased the yield of the plants. The berry mass also was enhanced by the 100% ETC treatment, while the total soluble sugars were decreased. This can be, as Dr. Castellarin explained before, this can be a result of the dilution that we observe when the berry size is increased. Regarding the secondary metabolites, we saw that in 2019, the flavonols and the anthocyanin contents in berry skins were dramatically decreased by the 100% ETC treatment, while no difference between treatments were recorded in the 2020 growing season. Now, I'm going to explain you a little bit what is the water footprint. The water footprint is defined as the total amount of water that we need per unit of product. And it is estimated as the sum of three components: the green water footprint, who came from the rain, that is used for this purpose; the blue water footprint, that is the water, the irrigation water that can be sourced from the surface or groundwater; and the gray water, that is the amount of water that we need to dilute the pollution to reach the quality standards for the water in for the drinking water. So we estimate this water footprint for our experiments, and we saw that the 100% ETC decreased the total water footprint, while the 25% increased it. And now I'm going to talk about the arbuscular mycorrhizal fungi. I don't know if you guys are aware of these fungi. They are natural soil symbionts that penetrate the cortical cell of the roots of the 80% of the vascular plants and form arbusculars. The latest research have shown that they are key components of the viticulture production systems because they provide ecosystem services provided by when they are in symbiosis with grapevines, like they increase the plant resistance against biotic stresses while reducing phytochemical input. They increase also the plant resistance to abiotic stresses such as drought, salinity, metals, and mineral nutrients depletion. Reduce the fertilizer requirement, improves the soil water retention, promotes the plant growth and the plant quality for human health, because it enhances the metabolism of many antioxidants in plants, and also improves the soil structure and the stability. So briefly, I'm going to explain to you how we analyze these fungi in the roots. So we sample the grapevine roots, clean them and stain them, and then we see them under the microscope. So you can see in the second slide the arbuscules produced by this symbiosis, and also the hyphae structures, and also the vesicles from the fungi. And then we quantify with these images, and we saw that the 100% ETC treatment decreased the arbuscular mycorrhizal fungi abundance, colonization rates, while the other treatment, the 25 ETC, increased it. So to conclude, we can say that the replacement of different fractions of crop evapotranspiration affect both primary and secondary metabolism, as well as yield components. The 25% ETC had a detrimental effect on yield components but improved berry quality. The 50% ETC treatment resulted in an improved berry quality without affected yield components, and the 100% ETC improved grapevine growth and yield but diminished berry quality through decreased total soluble solids in berries, anthocyanins, and flavonoids. So as a take-home messages, I want to say that water-deficit irrigation strategies must reduce the amount of water applied to grapevines, decreasing the water footprint, but also maintaining or increasing the grape quality at the cost of some reduction of potential yield. So after these 2-year trials, we can say that in spite of the warming trends and the lower precipitation patterns, the optimal irrigation still is still 50% or 60% ETC, because if not, we are decreasing the berry quality. So to finish, I want to thank all the people who work in this project, to the Dr. Can Cultural, Johan Martinez Lechers, Eumorphia Kostaki, and Ruth Yu, and also the financial support that we have for this trial. Here I left some reference that I used for this presentation. And thank you so much for your attention, and I will be really glad to answer to your question in the chat. Thank you.